Our next guest has leveraged technology and many other business innovations to quite literally change the world. Dr. Noubar Afeyan is founder and CEO of Flagship Pioneering, a company that conceives, creates resources, and develops first-in-category life science platform companies to transform human health and sustainability. An entrepreneur and biochemical engineer, Dr. Afeyan holds more than 100 patents and has co-founded and helped build more than 60 life science and technology startups during his 33-year career. He is co-founder and chairman of the board of Moderna, the pioneering messenger RNA vaccine company addressing the global COVID-19 pandemic, and he serves on the boards of several private and public companies. Dr. Afeyan, welcome. It's so good to have you.
Good to be here.
Thank
you. Thanks for having me.
Really looking forward to this conversation because I think your work touches almost every single one of the pillars of Signal, from technology to commerce to policy to corporate citizenship and beyond. So I'm looking forward to getting into this. I know everybody wants to hear about Moderna. I'm sure, if it's possible, you might even be sick of talking about it, but let's first talk about the company that helped create Moderna, which is your platform, Flagship Pioneering. What is Flagship and what makes it different?
Well, again, thanks for having me. Flagship Pioneering, now in its 21st year of existence, was really an experiment in how to create an institution that can make breakthrough innovations and also launch companies based on them. And we started with the notion that rather than kind of looking for innovations and trying to commercialize them, could we actually look for the needs and the big opportunities and make the innovations needed to have transformative impact? And how could you do that as a team? How could you do that as a company? So imagine a company whose product is a company or many companies, each one of them tackling different aspects of human health or sustainability. And that's what we set out to do. Flagship is highly predicated on the notion that breakthrough innovations can in fact be made systematically, but that they're not going to be made in the more traditional way where you follow the science and you incrementally try to find your way to that, but rather it involves a whole different way of thinking, and we call this emergent innovation. And using those approaches and several others, we've been able to, over the last 20 years, develop a whole methodology and a platform from which we're operating.
More about what emergent innovation is?
Sure. You could believe that breakthrough innovations are made by brilliant people, and there's no question that brilliant people are needed to make breakthrough innovations. But I would posit that breakthrough innovations are often emergent. And what I mean by that is that they are the result of a long series of iterations that at some point appear as a major gain of function, gain of capability, and that's what we deem a breakthrough. If you view it that way, then rather than trying to find these innovations in a fully made, fully formed way, you need to create an environment, a culture in which such emergence can happen. And that's really what we mean by emergent innovation.
Got it. Is there something about sort of the traditional approach to innovation, say in the pharma and health sciences industry, that make that kind of emergent innovation difficult?
Well, look, I think we need innovations of all types, and so I'm not here to say one is better than the other. It's just different. But indeed, traditional innovation is exploring adjacencies, right? So whatever seems like a reasonable next step and a next step after that is where people will try to create a balanced portfolio of investments and projects. Instead, if you want to get beyond those adjacencies, you're in a way in a white space, and it's not clear where you should go, and you need different mindsets. I'd say that the traditional pharmaceutical research establishment is rather more focused on the adjacency orientation because it's more reliable. You can use risk-reward analysis more predictably, Whereas this type of innovation seems a bit more kind of futuristic, but also hard to figure out which one is going to work, which one's not. And so I think people tend to avoid it.
Right. Let's get specific, because I think you've got, with Moderna, this 10-year overnight success, right? A company that for most of its life, most of the world had never heard of. And then almost within a month, everybody had heard about it, and it was in the news and has been in the news now, since the beginning of last year. Tell us the story of Moderna. How did it start? What was the technology behind it? And how does it fit into this pattern of emergent innovation?
Sure. So in 2010, where we had kind of been iterating this model for already a few years, as one of many explorations that we started, one of them basically was focused on whether we could think about a medicine that, when delivered to a human subject, could cause that human to make another protein, a protein that could be therapeutic. So the notion is can we use a delivered entity that can instruct the human body to make a given therapeutic protein? Now, how you do that, what technology you use to do that, was a bit open-ended. But during this exploration, the molecule class that really caught our interest was messenger RNA. Messenger RNA, it had been already reported in the literature, could be delivered into cells, but it causes a very strong immune response right out off the bat, which makes it somewhat useless as a drug. And that's where it stood for 15, 20 years since it was first attempted in the very early days of biotechnology. What had changed over the years, and we researched this to find out what the state of the art was that some chemical modifications could begin to tame the immune response. Not all the way, not enough to safely deliver to humans, but a bit of the way. Second, you need to get these into cells, and there was really no technology to get it into cells at the time. But we launched this project, we call this LS18, for the 18th life science project we had launched at that point, to ask the question, what if we could deliver medicines that caused humans to make their own drug? And started working on, okay, if you can chemically improve, can you do it much more potently? What are the ways you can get it into cells, muscle cells, liver cells? And after a year plus or minus of search, we found ways to actually in animals show that you could take an mRNA code, perfect it in its design, and these are things that we learned by doing, ultimately put it into some packaging and get it into mice and make a relevant protein. From there, you could look backwards and say it was engineering, meaning the proof of concept eventually would lead us not necessarily to every single protein drug, but some. That's where we started. Roll the clock forward 5 years, we, for the first time, tried this as a vaccine. It turned out we chose a vaccine back then because it takes 1 or 2 shots, and it's kind of almost the easiest technical challenge to show the proof of concept. And in fact, ironically, we went after 2 potentially pandemic flu strains. And I say potentially pandemic because, as you could imagine, in flu that varies every year, there are some strains that have been known to cause epidemics, and even some that could cause pandemics that have never entered into humans. And we thought that might be the safest thing to try out our technology against in order to see what kind of immune response we can get. Roll the clock to 2020, beginning of 2020, we had 9 vaccine trials that we had already done in humans, showing 9 different vaccine types, all with mRNA. And we were not expecting this coronavirus, but when it showed up, in hindsight, we were poised to go after it.
Yeah, that's a very fortunate choice that you made to focus on these areas so that you were ready when the time came.